Today we will discuss about backcross and test cross. When an organism expresses recessive characters, its genotype is surely known. For example, here dwarfness is a recessive character while tallness is a dominant character. So, the genotype of recessive character is surely known. It is small t. But it is not possible to know the genotype of dominant characters. It may be pure or it may be hybrid. For example, in monohybrid cross, F1 hybrids are definitely heterozygous, that is, capital T small t. But in F2 generation, all tall plants are not heterozygous. 25% are pure tall, 50% are hybrid tall, and 25% are pure dwarf. Therefore, to find out whether tall plants is homozygous or heterozygous, a test cross is carried out instead of selfing. Test cross is a type of backcross. Backcross. The cross of F1 individual with any one of the homozygous parent is called as a backcross. As the offspring is crossed back with one of the parent, it is called as a backcross. Homozygous parents are of two types: homozygous dominant parent and homozygous recessive parent. Dominant backcross. A cross between F1 hybrid with its dominant parent is called as a dominant backcross. Explanation. In a cross between pure tall pea plant and pure dwarf pea plant, the hybrid of F1 are all tall with genotype capital T small t. When F1 hybrid are crossed with homozygous dominant parent capital T, all the F2 individuals show dominant characters. That is, all the F2 individuals are tall in nature, which may be capital T or capital T small t. This is because homozygous or pure dominant tall parent will produces only one type of gamete, that is capital T, and hybrid tall capital T small t will produces two types of gamete, capital T small t. Therefore, all the F2 plants will have capital T in their genotype, as capital T and capital T small t, and individuals will tall. This cross will produces all tall, but 50% are pure tall and 50% are hybrid tall. Recessive backcross. A cross between F1 hybrid with its homozygous recessive parent is called as a test cross or recessive backcross. Its explanation. In a cross between pure tall and pure dwarf plant, the F1 hybrids produces are tall in nature, that is capital T small t. The F1 hybrid capital T small t are crossed with homozygous recessive parent small t. In the checkerboard method, the F1 hybrid, that is capital T small t, are crossed with homozygous recessive parent, that is small t. This produces two types of individual in F2 generation, that is tall and dwarf, in 1:1 ratio. This is because recessive parent produces only one type of gamete, that is small t, and hybrid will produce two types of gamete, capital T, and small t. So that, therefore, half of the progeny will have genotype capital T, small t, and half of the progeny have genotype small t. So, in the cross, the resulting progeny will be 50% tall in nature and 50% dwarf in nature. Thus, dominant and recessive are in 1:1 ratio. Such a cross is called as a recessive backcross or also called as a test cross. Significance of test cross. It helps to determine whether individuals with dominant characters are genotypically homozygous or heterozygous. Purity of parents can be determined. It helps to determine the genotype of individuals. It helps to introduce useful recessive character in hybrid in crop improvement programs. Deviations from Mendelian ratios. Many geneticists repeated Mendel's experiment by using other organisms and other characters. They observed many deviations or exceptions to the Mendelian ratios. This cannot be explained on the basis of Mendel's principle alone. This can be explained with suitable modification in Mendel's laws. Therefore, this is called as a post-Mendelian genetics or neo-Mendelian genetics or non-Mendelian genetics. They found lot of changes in the phenotype output, which was due to gene interaction. Gene interaction. The modification of the normal phenotype of certain genes by other genes is known as a gene interaction. Gene interactions are of two types. First, intragenic interaction, and second, intergenic interaction. First, intragenic interaction. This interaction occurs between the two alleles of same gene. It produces phenotype different from the normal dominant recessive phenotype. Common intragenic interactions are incomplete dominance, codominance, multiple alleles, and lethal genes. Intergenic interaction. This interaction occurs between the alleles of two or more different genes. The different genes may be present on the same or different chromosomes. They alter the normal phenotype. The intergenic interaction includes pleiotropy, polygenes, complementary genes, supplementary genes, epistasis, duplicate genes, etc.